Hello, everyone. My name is Darya Dayanim. I was in the Moore Lab under the mentorship of Dr. Lucas Derrick. My project revolved around silicon photomultipliers. You heard about them in previous projects, which are these photon detectors that are increasingly popular in physics experiments. And they're pretty much these array of mini photon detectors, which can only detect one photon at a given time. So once a photon hits it, there's a certain time interval where it also cannot detect another photon. This time interval, I'll be calling the reset times, and these mini photon detectors are what I like to call pixels. It's also important to note that the SIPMs are in Geiger mode, meaning they're biased at a point where one photon hits them and the same output signal will be given. So the motivation behind my project is that currently saturation models only exist for short and uniform light pulses that hit the SIPMs. So my goal is to characterize it for non-uniform light that hits it. So if we take, this is a very small segment of a SIPM kind of blown up. If we are currently able to index over every pixel and determine whether a pixel fired, meaning that it detected a photon hitting it. And if we kind of divide it in the X and Y and the Z, do some calculus and take the limit, we are able to come up with an integral. And this integral, very convenient for us, has an analytic equation for when a Gaussian beam is hitting the SIPM. My goal is, this is the model, not important. I can go over it happily more if someone wants to, but my goal is to see if conducting an experiment will show that my data will match this model. This x-axis is a varying flux of photons, and this y-axis is what the SIPM actually sees, how many photons it sees hitting it. This is a very simplified my optical path where I have a 405 nanometer laser, which will go through a pinhole to create a roughly Gaussian beam, right? Because we're working with a Gaussian model, so we want a Gaussian beam. And it'll go through neutral density filters, which is how I'm going to change the flux of the photons, which was the x-axis we saw in the previous graph. This laser will then go through a beam splitter where majority of the light will be directed to a calibrated photodiode, which is pretty much, it will tell me how many true photons hit it. And that's how we actually obtain the true number of photons on the x-axis. And then the rest of the light will be directed to the SIPM. The PD is mounted onto a manual XY stage just so that I can center it at the beginning of the trial. And the SIPM is on a motorized XY stage because that's how we're going to actually scan the SIPM. It's important to note that I'm using a laser because it's monochromatic, meaning one wavelength, and both the PD and the SIPM's responsivity is dependent on the wavelength. That's a picture of my actual setup. Okay, so this is a block diagram of my commission setup where I was just trying to make sure that my automated data acquisition works and that my linear stage was moving properly. This both entailed the code to make sure the movements were what I wanted it to be. There were also some mechanical issues that led to some inaccuracies in the movement that I wanted to deal with. So here we have my computer talking to an Arduino, which is controlling the stepper motors and moving the stage which the SIPM is mounted onto. We also have just a basic green USB laser pointed at the SIPM. This is a picture of my SIPM with its mount. And the SIPM is both biased and its current is measured by a picoammeter. This is some data with this previous practice setup that shows pretty much that I was able to get the data I wanted to, which this is a raster scan where every little pixel is, okay, I'm stopping at this point, drawing the current from the SIPM, moving 0.1 millimeter increments and going in a back and forth motion until I've covered the whole area of the SIPM. And these different colors represent how much current is being drawn. So it's obviously brighter in the center because the laser's straight onto the SIPM and there's more current being drawn. But it's not, we see how even though some less might be here, that's because of the saturation kink that we saw. These are also line scans, which I'm pretty much just going one across in the X and Y direction. And we can see this kind of, this error function being fit to it that will enable us to profile the beam, which is very important. And profiling the beam is the same as integrating the beam. And Ideally, we'd be working with the Gaussian, and the integral of the Gaussian function is the error function. But as we can see in the Store Labs diagram, this initial laser I was using is more of a top hat close up rather than a Gaussian from far away, which is why it's not fitting perfectly. This was my updated block diagram where the changes are, the main big changes I want you to see are in bold. So now we have the new laser diode, which will be roughly Gaussian leaving rather than top hat, and it's purple, or more blue, but it goes both to the photodiode and the SIPM through the beam splitter. The SIPM's current is now being measured by a digital multimeter and is being biased by some power supply. And then the photodiode's current is being measured by a picoammeter because its current is a lot smaller and is in need of a more sensitive device. This is kind of our final data where these are the scans that I showed before, and we can see how it fits a lot better of the error function. And these scans are very important because it gives us a standard deviation of the beam, which is needed in the model to actually see if the data fits. And then it also enables us, this dotted line represents a full width half maximum of the error function, and that gives us the dimensions of the beam. So we can see in this raster scan, the dotted blue line is what the computer notices to be the device's dimensions, which is in fact an accurate 6.1 by 6.1 millimeters. And this is kind of the star of the show, where we can see my data in the blue matches perfectly the model that we're trying to match it to, or seeing it if it accurately predicts it. This is just a refresher of the model and kind of where I got different parameters. I won't overwhelm people going through them. Again, happy to talk about it. And the rate of the photons is, just as a refresher, is taken from the PD's current with a changing, with the ND filters to change the flux. And the Y is taken from the SIPM in certain parameters such as gain taken from the data sheet that tell us how many photons it detected.
Sorry, just a 7-minute warning.
One more time?
Oh, a 3-minute warning.
Oh, sorry, thank you. And an important note is that the reset time, which is the delta T in this equation, which is the time interval I talked about where it can't detect another photon, is in fact a floating parameter because although it's given in the data sheet as 50 nanoseconds, this is not actually indicative of when a pixel will be able to detect another photon. So we left it as a floating parameter and it gave us about 268 nanoseconds. Oh. Okay, so summary, in case I lost you guys anywhere, I measured the SIPM saturation response to non-uniform light and it matched the model, which is pretty exciting. So my next steps are to implement a spatial filter pictured here in order to obtain a cleaner Gaussian because the model is supposed to work for a Gaussian beam. And the way it works is kind of shown here, where you take a collimated beam, you focus it down, and it gets rid of all the noise to output a perfect Gaussian. So that's what I hope to do, and then I'll have to align all the rest. And then I'd also like to test with what we call different over voltages, which is just changing the bias that's on the SIPM because we expect that to impact the reset time of the SIPM, and that will just let us test the tolerance of the model. And then finally, hopefully write up the findings. I'd like to give thanks to the Moore Lab. It was an incredible, supportive environment, and my mentor, absolutely amazing, Summer. Any questions?
So you said the data sheet said this delta T should be 50 nanoseconds and you measured 200. Factor of 4 seems like dramatically different. Like,
Okay, so the reset time, the recharge time that's mentioned on the data sheet is only obtained by the, here I can actually go to right here, is like a constant obtained by the quenching resistor times the capacitor in each cell.
Mm-hmm.
That's not something that's very much indicative of like what is actually the reset time. And we actually found that a capacitor to be fully recharged takes about 5 times that time constant, which would make sense with the 268 nanoseconds. But that's also why we want to test it with the different over voltages to see if that changes the reset time and if we get something that's astronomically not in range.
So I also tested zip pumps, which is there. When I tested zip pumps, I also used a blue light, and you mentioned that you ended up switching halfway through from a like green laser to a blue laser. And I know blue like light is a new thing, 2016 or whatever. What did you see as the actual like difference? Like why do we want to use the blue light?
So I used a blue laser, like a laser diode. From my understanding of is that what you used, a laser?
It was a light source, it was an LED.
Okay, yeah, so a laser is monochromatic, one wavelength just being outputted. So that was kind of an arbitrary choice. 405 is a cheap laser that we can get with a roughly Gaussian beam. The 405, the green laser, sorry, is what we just had lying around and what I could kind of commission the setup with. And a SIPM is blue light sensitive, so it's preferable to work with a blue light, but not a blue light in your case, like a laser diode blue light. So that was another reason to choose the purplish or 5 nanometer.
So when you do this raster scan, what's the size of single cell?
The size of, so that's not, okay, so let me break this down. Each pixel I take that I measure is 0.1 millimeters, but if you're talking about the actual cell of the SIPM, it's 35 microns, 35 by 35.
So when you do raster scan, you're trying to shine it on an individual cell or?
No, it's just kind of to see how the current changes as you get closer.
Okay.
Really, this is just a beautiful showing and to also show how the dimensions are accurate and we're taking the correct, but there's no significance as to one pixel being fired within the SIPM.
So the current you read is not necessarily from single cell.
It's
can be
not.
from bunch of.
Exactly, and the model actually depends on the beam being bigger than the cells in order for the calculus to work. So it's a 1 millimeter radius of a beam and the pixels are like 35 microns, so it's nowhere near.
Okay, thank you.